This video is meant to be a reminder of what variability is, perhaps a refresher from what you learned in a previous statistics course. So let's remind ourselves of what variability is. If we look here at these two samples or populations, this one is much more widely dispersed than this one, meaning that this has more variability than this one. So variability is a statistical measure, and it's going to describe the differences between scores. So how far is this score from the mean? So it's another way of thinking about it is the spread of scores or the distance of scores from the mean. So again, this score is closer to the mean than this score is. These scores are less spread out from the mean than these scores are. Large variability within a treatment, large differences are not going to be good. So differences that exist inside treatment conditions determine the variance of scores, and large variance in different groups can hide patterns in the data. So we want large differences between treatment groups, but small differences within a treatment group. And so another thing that variability does is measures the predictability, the consistency, and the diversity of score. So when you have smaller variance, you're going to have more consistency, more predictability. If you have larger variance, you're going to have a bigger diversity. Now, when we're talking about a between subjects design, large differences, like I said before, large differences between individuals could produce a large variance. So that's why we control for those variables. That's why we want our groups to be very similar to the sample and similar to each other. And we don't want to have large differences in individuals when we're measuring that dependent variable. So this could have a significant influence on the statistical interpretation or your significance. So significance means that it's very unlikely that the difference would occur if there was not a consistent difference between the treatment conditions. Or in other words, significant means that it's very unlikely that your results occurred by chance, that difference that you see between groups is real. However, if you have large variability within your control and experimental group, that could hide differences between those groups. So now we're just going to compare two 10-point effects and hopefully hit home what I was saying on the previous slide, if it didn't quite make sense. So here we have a 10-point difference. So the difference in mean between the control condition and the treatment condition is 10 points, between 40 and 50. Now you can see here we have a nice small variability here, going from 38 to 42. It's only a 4-point range. And same with here in our treatment condition. Now if we look down here, we still have a 10-point effect, but we can see our variability, now we're going from 20 all the way up to 60. We now have a 30-point range, and here, that's in our control condition. For some reason, the colors swapped in the two graphs. And then the treatment condition, we have a 36-point range. That huge amount of variability, so range is one way you can look at variability. That huge amount of variability doesn't allow us to see this 10-point effect at all. So in order to control for these individual differences that can influence variability, researchers can do a couple of things. One thing is to increase the differences between treatments. So here we have two treatments, and so here you see this is the mean here, this is the mean, and so this difference here, that's the difference between the treatments. We try to see if we can increase it. What can we do to increase that difference between the treatments? Then even if we have large variability within one of our groups or our sample, we can still see a real difference. The other thing is to decrease variance within the treatment. So here we have a group that has large variance and we want to decrease that. And some of those things that we talked about in the previous video, like matching or holding a variable constant, those are ways that you can do that. So now we're just going to talk about some ways we can minimize variance within our treatments. So one thing we can do is to standardize procedures and treatments. And we've talked a little bit about this before. You want your environment to be the same. If you're using a computer, you want it to be the same. If you're having your subjects listen to music, you want the volume to be the same. Standardizing those things so you don't have a huge amount of variance. Now with our experiments, it may not be as feasible to control some of these things, unfortunately. You want to try to limit individual differences. We talked about how we can hold variables constant, matching subjects, those types of things. So just briefly to discuss some ways we can minimize the variance within the treatments. So we can standardize procedures and treatment settings like we've talked about, making sure that the lighting is all the same when you're doing an experiment, making sure perhaps the same person's giving instructions. If you're having them listen to music, making sure that the volume on the headsets or from the computer is the same each time. We try to limit individual differences, and there's a variety of different ways we can do that. You can use random assignment, we can use matching, we can hold variables constant. And then sample size is also important. Now, if you have a gigantic sample, you're probably more likely to have a huge amount of variability because you have more individuals. And so we already talked about a good sample size is around 25 to 30 individuals. So you don't want to have too small a sample because then it may not be representative. So all of these 4 things are going to act to increase the internal validity. That means that the variability within your control and experimental group is small, and we can see real differences if they exist. Now, of course, there's going to be threats to internal validity. And one of these could be differential attrition. So this is the idea that there's differences in dropout rate from one group to another. So you recruit your participants before you actually get to participate in the study. People don't either complete it or drop out. And maybe it's in one condition versus another because one condition requires something different. So that differential attrition could create large differences between groups and create problems for you. Also, if you have any communication between the groups, so if the groups are communicating and they're letting each other know what's going on in the other one, then that can influence your internal validity as well.